So let's talk about semen. So why is there a need for the laboratory to analyze semen? So it is mainly because for fertility testing, post-vasectomy analysis, or the analysis if there is a successful operation of the removal of the vas deferens, as well as forensic analysis. So according to World Health Organization, the specimen, or rather the patient, must submit 2 or 3 samples. So the samples submitted must be collected 7 days to 3 weeks apart. Okay, so take note that at least 2 abnormal samples must be submitted or must be analyzed in order for it to be clinically significant. Okay, so what is the most common cause of male infertility? That is varicocele. So hindi mumps, okay? Varicocele. Varicocele is the hardening of veins that drain the testis. So this reason causes the blood from the adrenal vein to flow into the spermatic vein. So again, huwag kakalimutan, varicocele is the most common cause of male infertility. How about for the second reason? Si post-vasectomy analysis. As I've said earlier, vasectomy is the surgical removal of all or part of the vas deferens for the purpose of male sterilization. So yung kanina, fertility testing. Ito naman, to ensure na wala na makakapass through na germ cell or sperm cell. So in here, in the post-vasectomy analysis, the only concern is the presence or absence of sperm. So other tests for semen analysis like liquefaction time, sperm concentration or count, viscosity, uh, etc. So wala na siyang need for us to perform since ang only concern lang natin dito is yung presence or absence lang. So other variables such as temperature or storage, hindi na siya importante for post-vasectomy analysis. So kahit greater than 1 hour na siya masubmit sa laboratory, okay, okay lang. Okay, because our only concern here is the presence or absence of spermatozoa. So how will you perform post-vasectomy analysis? So the testing begins at 2 months post-vasectomy, and then you need to submit specimens monthly until 2 consecutive monthly specimens show no spermatozoa. Again, dapat walang spermatozoa for post-vasectomy analysis. So just in case there is a presence of a single sperm or single motile sperm, ibig sabihin nun, it indicates that there is a unsuccessful vasectomy. Okay, so again, wag kakalimutan, presence or absence of spermatozoa. How about for forensic analysis? So it is used for forensic analysis for suspected rape cases. So what are the tests that can be employed for forensic analysis? So you can either perform seminal stain for Florence and Barbiero, chemical analysis for PACP or prostatic acid phosphatase, PSA, or DNA analysis. Aside from that, you can also perform the microscopic examination of the specimen for the presence of sperm. Okay, so I will not be emphasizing these three anymore. Okay, so just remember that you can either use seminal stain, chemical analysis, or microscopic analysis. Next, we have yung semen physiology natin. So the semen is composed of 4 fractions coming from different organs. So it either comes from the testis epididymis, seminal vesicles, prostate gland, or bulbourethral gland or Cowper's gland. So as you can see here in the table, the spermatozoa, the germ cell itself, contributes the little volume, about 5% only. So makikita naman natin dito sa seminal vesicle contributes the majority of the volume of the specimen, about 60 to 70% of the specimen. Okay? So, isa-isayin natin per organ kung ano yung mga pinoproduce or sinisecrete niya in the semen. So for testes, spermatozoa are secreted in the seminiferous tubules present in the testis. So seminiferous tubules is the organ in the testis that secrete spermatozoa or the germ cell. So the spermatozoa are attached in a Sertoli cell, which is considered as the nurse cell because it provides support as well as nutrients to the spermatozoa as they undergo development. So let's discuss the normal development of the spermatozoa or spermatogenesis. So it begins at type A spermatogonia. So type A spermatogonia is like the pluripotential stem cell because it replicates and dito nagsa-start yung maturation natin. Okay? So type A spermatogonia doesn't participate in the maturation. So ang ginagawa niya lang is to replicate itself. Okay? And some parts or some type A spermatogonia will develop into type B spermatogonia. So from type B spermatogonia, it develops into primary spermatocyte and then into secondary spermatocyte, into immature spermatozoa or spermatid or spermatozoa or yung mature na. Okay, so what is the difference between an immature spermatid or the mature spermatozoa? So ang main difference lang yan is that si spermatozoa or mature spermatozoa may presence ng motility. So kung may presence ng motility, si mature spermatozoa, it already has flagella. Okay, so saan ngayon nakukuha ni immature spermatid yung flagella for it to be considered as mature spermatozoa? So it gains its motility and flagella at the organ epididymis. So in the epididymis, the sperm mature and develop its flagella. So it is the place wherein they develop their motility. So the entire process of the development of flagella approximately takes 90 days. Okay, 90 days. So aside from that, ano pa yung function ni epididymis natin? It also serves as a storage of spermatozoa until ejaculation. So until needed, si spermatozoa natin, nakatambay lang siya sa epididymis natin. So if si patient, masturbation or sexual intercourse, okay, from the epididymis, it will be propelled to the ejaculatory ducts through peristaltic action. Okay, next we have the seminal vesicle. So as I've said earlier, seminal vesicles produce seminal fluid, which produce the majority, about 60 to 70% of the normal volume of the semen. So ano yung laman ng seminal fluid natin? So it contains a high concentration of fructose, so sugar, and flavin. So flavin is the pigment responsible for the grayish coloration of the specimen or the semen. So spermatozoa metabolize the fructose for energy and needed for the flagella to propel themselves through the female reproductive tract. So for example, there is a problem in the semen volume or the specimen volume. So kung mababa yung volume ng specimen, you can attribute it to the problems arising from the seminal vesicle. Okay, so for example, masyadong konti yung specimen. So baka may problem si patient sa seminal vesicle. So one way you could assess if there is a pathology in the seminal vesicles is by assessing the fructose level. So the assessment of fructose level will be discussed later. How about for the prostate? So prostate is a muscular gland located below the bladder. So it secretes acidic fluid, contains high concentration of ACP, citric acid, zinc, and proteolytic enzymes. So para mas madaling isipin, si prostate is an acidic fluid, okay, acidic fluid, and it is responsible for the normal coagulation and liquefaction of specimen. So for example, after 1 hour, hindi nag-liquefy yung specimen. So you can attribute it to the problem in the prostate gland. Okay? Okay, for example, ang may problem naman is yung pH naman ni specimen. Okay, for example, masyadong alkaline. Okay, masyado namang alkaline. So, kung masyadong alkaline, again, baka naman walang acidic fluid. So, again, sino may problem? Si prostate gland. Okay? How about for bulbourethral gland? So bulbourethral gland or Cowper's gland is located below the prostate. So what does it secrete in the semen? So it secretes a thick alkaline mucus of fluid that helps to neutralize acidity from the prostate secretion as well as protection from the male urethra and the vagina. So if you try to remember, the vagina is acidic in nature. So again, di ba kung maalala, may bacteria dyan that maintains its acidic environment, which is the Lactobacillus acidophilus. How about for male urethra? So acidic rin siya because, of course, the presence of urine. So again, isa lang naman yung daanan ng semen natin as well as yung urine. Next, nandito naman na tayo ngayon sa specimen collection. So before submission of the specimen, the patient must ensure that he did not perform any sexual activity such as sexual intercourse or masturbation. So prior to the submission of the specimen. So dapat 2 to 7 days siya sexual abstinence. Okay? 2 to 7 days or 2 to 5 days. So the specimens collected following prolonged abstinence or greater than 7 days tend to have higher volumes or decreased motility. So decreased motility because some of the sperm was already dead. Okay? If there is a prolonged abstinence. So all portions of the ejaculate must be collected because if there is a certain portion missing, so makaka-affect yung sa result natin. So for example, first portion is missing. So ano ba yung laman ng first portion? So it contains majority of the germ cell itself with an addition of the prostatic secretion. Again, if you try to remember, prostatic secretion is acidic in nature. Okay, as well as it is responsible for the normal coagulation and liquefaction of the specimen. So kung wala yung first portion, ano yung ma-observe natin na changes in the tests? So decrease in sperm count again, kasi wala yung majority ng spermatozoa. pH is falsely elevated or alkaline because again, wala na siyang acidic secretion coming from the prostate and the specimen will not liquefy. So again, walang prostatic secretion, hindi mag-liquefy or magkakoagulate yung specimen. How about for the last portion? So, paano kapag walang last portion? Ano ba yung laman niya? So, ang laman niya, mainly is seminal fluid. Seminal fluid, again, is the responsible for the normal or 60 to 70% of the normal volume of the specimen. So, paano kapag wala yung 60 to 70% na yon? So, of course, tataas ngayon yung sperm count mo. Okay, tataas because maging concentrated yung germ cell. Kasi wala yung transport medium, which is yung seminal fluid. How about yung pH? pH is falsely decreased because seminal fluid is alkaline in nature. How about for semen volume? So again, 60 to 70% of the normal specimen volume is because of the seminal fluid. So bababa ang semen volume. Okay. Next. So specimens should be collected by masturbation. So if hindi possible, so dapat si patient gumamit ng non-lubricant containing rubber or polyurethane condoms. So these condoms kasi wala silang spermicide. Okay? So it must be collected in a warm sterile glass or plastic container. So usually, ang recommended na laboratory is that yung collection within the laboratory itself lang or may separate rooms in the laboratory for collection. So just in case hindi pwede or walang area for collection, so specimen must be transported to laboratory within 1 hour of collection and must be maintained at room temperature. So for example, na-receive na yung specimen. So, na-receive na yung specimen and may mga iba pang specimens na i-analyze. So, yung specimen mo, pwede mo siyang i-store at 37 to preserve its viability. So again, before coming to the laboratory, kept at room temperature, but if it's already in the laboratory and there are specimens that are being analyzed before the semen, so you can store it at 37 degrees Celsius. So now we will discuss semen analysis itself. So you must remember that in semen analysis, it should be performed generally after liquefaction. So ibig sabihin, dapat at least 30 minutes or 60 minutes must have passed before mo siya i-analyze. Okay? Again, so all tests for semen analysis must be performed after liquefaction time. So after 30 to 60 minutes. So if, for example, yung specimen natin, hindi siya nag-liquefy after 2 hours o masyado mahaba. So you can add phosphate buffered saline or alpha chymotrypsin or bromelain. So alpha chymotrypsin or bromelain are proteolytic enzyme. Okay, to liquefy or to forcibly liquefy the specimen. So usage of these treatments must be documented because this proteolytic enzyme may interfere with certain tests. Okay, so it may interfere with pH, okay, or the counting itself. Okay, so usage of these treatments must be documented. How about for the appearance? So normal appearance of the semen is gray-white, translucent, and musty odor. So again, the gray-white coloration is attributed to the flavin secreted by the seminal vesicles. So how about if there is an increased white turbidity? So it indicates infection. Again, infection, of course, presence of WBCs. So the presence of WBCs in semen must be differentiated from immature sperm. So why is there a need for you to differentiate the WBC against immature sperm? Because they are similar in morphology. So pabilog kasi sila. Okay, round yung morphology nila. Okay, so what if there is a red-brown coloration? Of course, presence of blood, yellow coloration, urine contamination, specimen collection, following prolonged abstinence and medication. Take note that if it's contaminated with urine, reject na agad yung specimen because urine is toxic to the sperm. Okay, so it must be rejected. Next, we have yung normal volume. So normal semen volume is 2 to 5 ml. Okay, so again, if you try to remember what is the organ responsible for the majority of the volume of the specimen. So again, it is attributed to seminal vesicles. So if there is an increased volume, prolonged abstinence. If there is decreased volume, again, nasabi ko kanina, baka may pathology arising from the seminal vesicle. So aside from that, you could also consider if there is an incomplete specimen collection. So again, ano yung portion na wala? For example, kung decreased yung volume ng specimen. So again, wala yung last portion. So wala yung last portion. Next, how about viscosity? So viscosity refers to the consistency of the fluid and may be related to specimen liquefaction. So the normal viscosity of the sperm is that after drawing into a pipette, it must form discrete droplets. So again, normally after 60 minutes, dapat hindi na viscous yung specimen natin. Dapat runny na siya. So dapat it is able to form droplets after 60 minutes. Dapat wala nang appearance ng string or clumpy. Okay? Take note again, dapat droplets na lang. So for example, na-notice mo, meron pa ring string part or stringy part or clumping. Okay, so of course, increase na yung viscosity. So again, what problems or what organs is associated with increased viscosity? So it is attributed or maybe are attributed to problems in the prostate gland, which is responsible for the normal liquefaction and coagulation. So increased viscosity may impede testing for sperm motility, sperm concentration, anti-sperm antibody detection, and measurement of biochemical markers. So the grading for viscosity is 0 for watery, 4 for gel-like. So, ang palatandaan ko dyan, yung 0, mukha siyang water drop. Okay, water droplet. Okay, so 0 for watery, 4 for gel-like. Okay, next, we have yung pH. So, pH should be measured within 1 hour of collection. Normal pH is 7.2 to 8, slightly alkaline. Increased pH indicates infection within the reproductive tract, of course, with the presence of bacteria. Decreased pH associated with increased prostatic fluid, ejaculate duct obstruction, or poorly developed seminal vesicle. So again, prostatic fluid. So again, prostatic fluid is acidic in nature. That's why mas lalo siyang maging acidic. Okay. So again, you can also consider, or should also suspect. You should also suspect that certain inadequacies in the specimen collection. So, pwedeng yung first or last portion. Ano kaya yung pwedeng wala? Saan ba yung pwedeng magkaroon ng decreased pH? Again, yung first portion natin, ang laman nun is the majority of the spermatozoa and prostatic secretion. Okay, so yung last portion, wala naman yung seminal fluid. So, in here, for decreased pH, you should also suspect that there is an inadequate specimen collection. Okay, wala yung last portion. Okay, wala yung last portion. Okay, wala yung last portion. Next, we have sperm concentration. So even though fertilization is only accomplished by one spermatozoa or one spermatozoon, the actual number of sperm present in the specimen is a valid measurement of fertility. So in layman's term, para more chances of winning. Okay. So what is the traditional lutein fluid used in sperm concentration? Sodium bicarbonate or formalin. Pero usually in the laboratory, good results may be obtained by using just ordinary uh, saline or distilled water. Okay, so what is the counting chamber being used here in sperm concentration? So we usually use Neubauer counting chamber, but you can also use Mackler counting chamber. So the main difference between Neubauer counting chamber and Mackler counting chamber is that for Neubauer counting chamber, you can only perform sperm concentration here. But for Mackler counting chamber, you can use it for both determination of sperm concentration and sperm motility. Okay. Next, the sperm concentration, the normal value is greater than 20 million per ml. The borderline result, 10 to 20 million per ml. So again, may formula ulit tayo dito for sperm concentration. So again, itong formula natin for general cell counting, cells counted times dilution factor over airs counted multiplied to death. Okay? But in here, dito kay sperm concentration, don't forget, after getting the result here, you must multiply the answer to 1,000. So ano yung 1,000 na yan? That is the factor to convert the cubic millimeter to ml. Okay, kasi yung ml, ito yung unit natin dito for sperm concentration. So the number of sperm per ml. Okay, so wag kakalimutan, for sperm concentration, the unit is million per ml. So both sides of the hemocytometer must be counted, and if since dalawa yung count mo, so both sides, it must be averaged. So the counts should agree within 10%. So if the counts do not agree, dilution and the counts are repeated. So take note here for sperm concentration, only fully matured sperms are counted. So what do you mean by fully matured sperm? So again, those sperms that have flagella. Okay. So how about for immature spermatids and WBCs? So again, immature spermatids doesn't have any flagella. So of course, hindi siya kasama sa counting, as well as WBC. So si immature WBC and spermatids, heads up ko lang, these are what we call as round cell. Okay? Si immature spermatids and WBCs, these are what we call as round cell. So mamaya, i-discuss din natin siya. For sperm count, you can derive sperm count by multiplying the sperm concentration, which again, unit niya, million per ml, to the volume of the ejaculate. So here, for sperm count, it is highly dependent on the volume of ejaculate. Okay, so again, kung maaalala, sinong organ responsible for the normal volume of the semen or the specimen? Again, yung seminal vesicles. So if there is problem in the seminal vesicles, again, it may interfere or it may affect yung result natin for sperm count. Okay, so yung normal value natin dito, 40 million per ejaculate. So yung unit naman natin dito, ejaculate. Okay, unit natin is ejaculate, million per ejaculate. So how do we arrive greater than 40 million per ejaculate? If you try to remember, yung normal result natin for sperm concentration is 20 million. And the normal result naman for specimen volume is 2 to 5 ml. Okay, so multiply yung 20 million sa 2 ml. So that's how we arrived at greater than 40 million per ejaculate. So here is an example kung paano tayo magko-compute for semen concentration and count. So using a 1:20 dilution, 34 and 38 sperm cells are counted in the 5 RBC counting squares on both sides of the hemocytometer. Calculate the sperm concentration and the sperm count with a volume of 4 ml. Okay, so before we answer this, so check muna natin yung mga given. So again, may dilution na given. So may cells counted. So may specimen volume. Again, walang given na area. Ang sinabi lang dito, again, is 5 RBC counting squares. Kung maaalala natin, ano ulit yung area if nabanggit si 5 RBC counting squares. So again, ang area natin for 5 RBC counting squares is 1 over 5 or 0.2 square millimeter. So ang formula again, cells counted multiplied to dilution factor over areas counted multiplied by 0.1 millimeter. So again, yung cells counted natin, dalawang given, it should be averaged. Again, dilution factor, ano yung reciprocal ng 1:20 dilution? 20. Yung areas counted naman, again, for 5 RBC counting squares, 1 over 5 or 0.2 square millimeter. Multiplied by the depth. Again, yung depth natin, constant yon, which is 0.1 millimeter. So, is equivalent to 36,000 cubic millimeter. So again, final answer na ba dun 36,000? Okay, hindi. So 36,000 is multiplied to 1,000, diba? To convert the cubic millimeter to ml. So you will have 36 million per ml. So is this the final answer? So this is not the final answer because it is the question is asking for the sperm count. Okay, so you need to multiply 36 million per ml to the specimen volume. So 36 million per ml multiplied to 4 ml. So the answer here is the 144 million per ejaculate. So this is the sperm count. So later we'll be having questions, so I hope you can answer it. Next we have sperm motility. So sperm motility should be assessed using a well-mixed liquefied semen. So again, lahat naman ng testing within semen analysis dapat fully liquefied ang semen within 1 hour of specimen collection. So lahat naman ng test here for semen analysis must be performed within 1 to 2 hours of specimen collection. Okay, don't forget, all testing for semen analysis must be tested within 1 to 2 hours of collection. How will we evaluate sperm motility? So it is estimated after examination of 20 high power fields, or alternatively, you can also evaluate 200 sperms per slide. So at least 20 high power fields or 200 sperms per slide. Traditionally, ang ginagamit natin sa laboratory is that we will estimate by means of percentage, yung motile, and then with an addition of yung criteria depending on sa speed and the direction. But according to WHO Laboratory Manual for the Examination and Processing of Human Semen 2010, so imbis na speed at direction, ang nire-require na lang ng World Health Organization is direction only. So wala nang kasama na speed. So ano ba yung criteria natin traditionally as well as in WHO? Traditionally, ito yung grading natin. So again, if you try to observe here, there is an evaluation of speed, kung rapid ba siya, slow, and of course, there is a presence of also the direction. Straight line, lateral ba siya, may noticeable lateral. Okay, so the normal result here for the additional grading is that there should be presence of greater than or equal to 50% motile sperms with a rating of 2. If you try to observe the grading of 2, it means that there is a presence of slow forward progression with noticeable lateral movement. Again, 50%, greater or equal to 50% motile sperms with a rating of 2 after 1 hour. How about for the published guidelines by the World Health Organization? So according to WHO, again, there is no presence of speed grading. So puro direction na lang to. So again, kung ma-observe nyo, sperm moving linearly or large circle. Okay, so wala na siyang speed, wala nang rapid or slow. Okay, so how about for WHO? The normal result naman is greater than or equal to 25% motile with PM grading. So progressive motility grading, sperm moving linearly or in a large circle. Sperm moving linearly or in a large circle. Next, how about for sperm morphology? So sperm morphology is evaluated with respect to the structure of the head, neck piece, mid piece, and tail. Of course, the sperm anatomy. So problems arising from the head is associated with poor ovum penetration. How about for neck piece, mid piece, and tail? So all of these are pathologies rising from neck piece, mid piece, or tail may influence the sperm's motility. So madali lang naman, head, poor ovum penetration, lahat, motility. So I will not be emphasizing this, but I just want to say again, head is critical for ovum penetration because of the presence of the acrosomal cap. So the acrosomal cap should encompass 1/2 of the total sperm head and 2/3 of the sperm's nucleus. Okay, so again, head is critical for ovum penetration. How about for midpiece? So midpiece again, problems arising from the midpiece may affect motility. So it contains the mitochondrial sheath, which is considered as the thickest part of the tail. Next, how about the tail? Again, it is responsible for the sperm's motility. Saan again ito nade-develop? Okay, nade-develop ulit ang flagella natin or tail sa epididymis. So how will you evaluate sperm morphology? So it is evaluated by using thinly smeared stain slide using oil immersion. So just like when reading or evaluating RBCs, you should use oil immersion field. So air-dried slides or slides are stable for 24 hours. So kahit may time na nagpass, still same pa rin yung result. So at least how many sperms must be evaluated for morphology? So imbis na 100 sperms for morphology, at least 200 sperms. At least 200 sperms should be evaluated and the percentage of the abnormal sperm must be reported. So again, huwag kakalimutan 200 sperms must be evaluated for normal morphology. Next, so these are the common sperm morphological abnormalities. So again, this normal head, so double head, giant head, amorphous head, pinhead, taper head, constricted head are the normal common head abnormality. For the common tail abnormality includes double tail or the coil tail. So again, spermatids, okay, dito kasama sa counting ng sperm concentration. So again, wala siyang presence na tail. So this is an example of spermatozoon with bent neck. So here is an example of a sperm with bent neck. So bent neck sperm is associated with head neck piece rather abnormally long neck piece abnormality. So abnormally long neck piece abnormality with bent head. So ano yung magiging excuse So ano yung magiging problem natin if there is an abnormally long neck piece and bent head? So it may interfere with motility. Next, how will we evaluate sperm morphology? You can either use the routine criteria or the traditional criteria and the Kruger strict criteria. So the routine criteria just evaluates the presence or absence of the morphological abnormalities. So for example, kung may double tail, double head, ah, ano pa ba? Tapered head, amorphous head, yung mga yan, yung mga napinggit natin kanina. Okay, so the normal value for the routine criteria is that there should be greater than 30% of the normal forms. Okay, how about for Kruger's strict criteria? So just by the name itself, strict, aside from the evaluation if there is a presence or absence of morphological abnormalities, you should also measure the head, neck, and tail acrosome size. So in here, yung difference niya, may measurement pa ng mismong head, neck, tail, or acrosome size. So kung masyadong malaki yung head, masyadong maiksi yung tail, masyadong mahaba yung neck, okay, there, it is considered as an abnormality in Kruger's strict criteria. Aside from that, presence of vacuoles must also be noted for in Kruger strict criteria. So how will you evaluate the sizes of the head, neck, tail, and the midpiece or neck piece? So you can use micrometer or morphometer, okay, to measure the sizes of those parts. Okay, for Kruger strict criteria, the normal value is that the sperm or the specimen should have greater than 14% of the normal forms. So this is the criteria recommended by the World Health Organization. So may measurement ng mga sizes ng mga part ng sperm. How about for round cells? So again, as I've said earlier, in sperm concentration, you should only count the fully matured sperms. So you will not count the presence of WBCs as well as immature spermatids. So again, WBCs natin and immature sperms or spermatids are what we call as round cells. So hindi siya kasama sa sperm concentration. But it must be noted and evaluated because problems may arise if there is an increase in WBCs, such as infection, or increase in immature sperm, such as in cases of abnormal spermatogenesis. So how will you differentiate WBCs and immature sperm or spermatids? So you can differentiate them by means of peroxidase stain. So again, if you'll try to recall in hematology, peroxidase or myeloperoxidase is considered as the primary granules of the granulocytes. That's why in peroxidase stain, ang magpa-positive is si WBCs. Positive si WBCs when compared with immature sperm or spermatid. So the peroxidase positive granulocytes is the most predominant form of leukocyte in semen. So again, because granulocytes are the WBCs that have myeloperoxidase in their granules. So again, si lymphocytes, again, walang granules yon. How about for computer-assisted semen analysis or CASA? So, kung ma-observe nyo, halos lahat ng mga tests natin for semen analysis, they are highly subjective. So why? Why is that considered they're highly objective? Because it highly depends on the proficiency of the medical technologist. So, depende yon sa kung paano basahin ni MedTech. Okay, for example, si MedTech, para sa akin, rapid yung movement ng specimen. Para naman sa isa, mabagal. Sa akin naman, may forward progression. Sa kanya, walang forward progression. Okay, so it is highly subjective. So nowadays, there already developed a procedure that is much more objective. Okay, but this procedure is found primarily only in laboratories that specializes in audiology. So ito yung tinatawag natin na CASA or computer-assisted semen analysis. So for CASA, it includes tests for sperm morphology, concentration, trajectory, and velocity. So for trajectory and velocity, basically, ito yung sperm motility natin. Trajectory, ito yung direction, velocity, yung speed. Okay, so basically, sperm motility, concentration, and sperm morphology. So yung mga routine semen analysis natin. Next, we now go to the additional tests. So again, as I've said earlier, there are tests that can only be performed in specialized antigen laboratories. So kasama na dun si CASA and itong mga additional tests na to. So if there are problems in the routine semen analysis, you can further the examination by performing these tests. So it includes sperm vitality or viability, anti-sperm antibodies, or microbial testing. So again, may problem So again, may problem na tayo sa routine semen analysis and you're just trying to further the cause kung bakit nagkaroon ng ganong problem. Okay, so for example, you've encountered that normal naman yung sperm count ni patient but you've noticed that in the sperm motility, masyadong mababa. Okay, less than 50% yung motility, for example. So, ano kaya yung pwede mong gamitin in order to further yung cause kung bakit hindi gumagalaw yung mga sperm natin? Okay, so you can perform sperm viability or sperm vitality. So how will you evaluate sperm viability or sperm vitality? By performing modified Bloom's test or the eosine or nigrosin stain. So basically, sperm vitality is nitrogen stain is a stain for the viability of the sperm, whether if the sperm is dead or alive. So it must be assessed within 1 hour of collection. You should count the number of dead cells in 100 sperm cells using bright field or phase contrast. So ang bibilangin mo dito sa AUC nitrogen stain is yung patay. So ano ba itsura ng mga patay in AUC nitrogen stain? So the not dead cells in AUC nitrogen stain are colored red against purple background. Meanwhile, for viable cells, ang color naman nila yung bluish white, yung normal na color. Okay, so ang basic principle kung bakit yung mga patay siya yung may stain is that when the sperm die, yung membrane ng mga sperm nagiging porous. If it's become porous, they are able to uptake the dye. Okay, so kaya na-stain yung mga patay natin na sperm cell. So for viable cells, since hindi sila nagiging porous yung membrane nila, so it retains its original color which is bluish white. Okay, so the normal result here for sperm vitality is that there should be a presence of greater than or equal to 50% viable cell. So, ibig sabihin, greater than 50% yung walang dye. And should correspond with evaluated motility. So, what if, yun nga, na-mention natin, pinaperform natin to, if there is a decreased motility. So, paano kung may problem sa eosinophil stain? So, ibig sabihin, maraming patay, kaya maraming hindi gumagalaw. So, that should be attributed to epididymal pathology. So, if you try to recall, before ejaculation, saan tumatambay yung mga sperm natin? Okay, so again, they are stored at the epididymis. So, ibig sabihin, kung by ejaculation, na-notice mo na hindi na gumagalaw or non-viable na siya, baka mismo yung epididymis or storage na yung may problema, which is the epididymis. So what if, again, normal naman yung nigrosin natin or sperm vitality, still decreased yung motility natin. So it can be attributed to flagellar abnormality. Baka naman yung sperm may abnormality na yung sperm ay yung flagella ng sperm. So baka masyadong maikse, okay, or other conditions. Next, what if there is a decreased sperm count? What if there is a decreased sperm count? Again, if you try to remember, sperm count is highly dependent on the specimen volume. So again, which is responsible for the majority of the semen volume? That is again seminal vesicle. So if there is a problem in seminal vesicle, there will be a lack of support medium, which is the seminal fluid. Okay, so how will you evaluate if there is a problem in the seminal vesicle? You can perform seminal fluid fructose or by means of resorcinol test. So resorcinol test produces an orange color when fructose is present. So resorcinol is a reagent that stains or that detects pentose sugar, which is here in this case fructose. Okay, so it produces an orange color. So the normal result for resorcinol test is that there is a presence of greater than or equal to 13 micromole per ejaculate. So it is determined by means of a spectrophotometric analysis. So it must be tested within 2 hours or collection, but if there is a delay in testing, you should freeze the specimen. Okay, it must be frozen to prevent fructolysis. So what if there's a problem in the resorcinol test? So it only means that there is a low fructose level. So again, if there is a low fructose level, there are pathologies arising from the seminal vesicle. So aside from that, it can either be problems with bilateral congenital absence of vas deferens, retrograde ejaculation, or androgen deficiency. Next, we have decreased motility with clumping. So you notice that after in liquefaction, na-notice mo na it doesn't form droplets, but it appears stringy or clumpy. So possible abnormality is that may presence na male anti-sperm antibodies. So, how will you evaluate male anti-sperm antibodies? By performing mixed agglutination reaction or immunobead tests. For mixed agglutination reaction, so, or MAR, it is a screening procedure and it is used primarily to detect the presence of IgG antibodies. Okay? So, yung sperm natin coated by IgG antibodies. So, how will you perform mixed agglutination reaction? You need to incubate the specimen with monoclonal AHG or anti-IgG. After which, you should add latex particles or IgG-coated RBC. So the latex particles or IgG-coated RBCs act as indicator cells to visualize the reaction. So latex particles or IgG again acts as a visualizing agent. So after performing that, so a normal result dito is that less than 10% should be agglutinated. So less than 10% yung agglutinated. So again, mixed agglutination reaction, one type of antibody lang yung madedetect niya, only IgG antibody. How about for immunobead test? For immunovid test, it is more specific than mixed agglutination reaction since it detects the presence of IgG, IgM, or IgA antibodies. Earlier, I've said for mixed agglutination reaction that it only detects IgG. For immunovid, it detects IgG, IgM, or IgA antibodies. So, mas specific siya. Aside from that, it can also demonstrate what part of the sperm yung target ng autoantibodies na yon. So, here in this picture, for example, this is an example of an immunobead. So this is an antibody to IgA, so IgA antibody. So saan siya naka-attach? Of course, sa IgA present in the head of the sperm. So basically, in this patient, for example, si patient may presence ng IgA head antibody. Okay, for example, ito naman, IgA antigen daw to, IgA antigen, and then nag-attach daw yung IgA. So, ibig sabihin, itong immunovid natin, naka-attach sa IgA again, still same pa din, but iba lang yung part na target ng sperm. Target sa sperm. So, this immunovid targets IgA tail antibody naman. So, the presence of beads on less than 50% is considered the normal defined by the World Health Organization. So, kanina, less than 10% yung agglutinated cells. For MAR, for immunovid naman, less than 50%. Next, you can also perform sperm agglutination with male serum to detect autoantibodies. How about if still normal naman lahat, normal yung routine analysis and other tests, but still, di pa rin magkaanak yung couple. So, you should suspect or consider that there is a presence of the female anti-sperm antibody. So, how will you detect or evaluate female anti-sperm antibodies? So, you need to perform sperm agglutination with female serum. So, the sperm or specimen is incubated with female serum. Next, last natin for microbial testing. So, greater than 1 million WBCs or again, increased WBCs indicate infection. So, what are the common bacterial causative agents that may cause infection in the sperm or the male reproductive tract? You can test for the presence of Chlamydia trachomatis, Mycoplasma hominis, Urea plasma, Urolithecum. Sila yung most frequently na dinedetect, okay, for semen microbial testing. Okay, so that's it for your discussion. So we'll now have the questions regarding what we've discussed earlier. So a patient who had vasectomy 2 months ago will send a semen sample for post-vasectomy analysis. It was collected in a plastic container and was left in room temperature for 1 hour before being sent to the laboratory. What will you do next? So again, ang reason, if you try to remember earlier, again, one of the reason for the analysis of semen is mainly due to post-vasectomy analysis. So again, for post-vasectomy analysis, our only concern here is the presence or absence of the sperm. So other tests such as liquefaction time, pH, motility, concentration, so wala na tayong concern doon. So ibig sabihin, variables or factors such as time, temperature of collection, hindi na natin tinitake note. Okay, so ang concern lang naman natin again is the presence or absence of sperm. So for this question, the actual answer is letter D. So again, for post-vasectomy analysis, hindi natin concern ang temperature at time of collection. So again, ang concern natin here for post-vasectomy analysis is the presence or absence of sperm. So the answer to this question is letter D, accept the specimen. Next, presence of fructose in resorcinol test will result in what color? The answer is letter B, orange. So again, resorcinol is a test for seminal fluid fructose. So the answer is orange. Which stain is used to measure for sperm viability? So again, sperm viability is performed if there is a normal sperm count with decreased sperm motility. So which stain will evaluate sperm viability or vitality? So the answer is letter A, eosin nigrosin. So again, eosin nigrosin stain will stain the dead cells. So the dead cells will stain red against purplish background. Next, a sperm count is diluted 1:20 and 50 sperm are counted in two large squares of the Neubauer counting chamber. The sperm count in ml is... So again, may computation tayo. So again, if you try to remember, ano yung formula natin? Cells counted multiplied to dilution factor, areas counted multiplied to 0.1 millimeter. So again, ilan yung sperm niya na nabanggit? 50. Okay, so there is no need for average, 50. So again, yung dilution factor given naman, 1:20. How about for areas counted? So in here, kung mapapansin, hindi 5 RBC squares yung gagamitin. So ano magiging area niya? So if you try to remember yung Neubauer counting chamber area natin, so this whole counting area, this whole area is 9 square millimeter yung total area niya. So kada square, kada secondary square, okay, kada secondary square is considered as having 1 square millimeter area. Okay, so 1 square millimeter area. So, di ba usually in hematology, dito tayo nagbibilang ng WBCs, di ba? Sa apat na corners. Sa RBC naman dito sa gitna. Di ba? Again, sa gitna, central square, central secondary square, and then sa apat na corner and sa central square. So, ito yung, again, yung sa RBC, 0.2. Ito naman, again, 1 millimeter. So, again, if you try to remember, 1 millimeter yung secondary square. So, dalawang large square daw. Okay, so ibig sabihin, dalawang secondary square na ang area ay 1 millimeter. Okay, so ibig sabihin, two large squares is equivalent to 2 square millimeter. So, multiply to depth, 0.1. So, maging answer mo is 5,000. So, again, 5,000 is not the final answer. You still need to multiply it with 1,000. So 5,000 times 1,000 is 5 million per ml. So the answer is 5 million per ml. So the answer here is letter D. When evaluating sperm motility, blank high power fields or blank sperms can be used. So again, for sperm motility, at least 20 high power fields or 200 sperms can be used. Following a normal sperm motility test with normal sperm count, what additional test might be ordered? So again, normal yung sperm count, but there is a decrease or abnormal sperm motility. So you can check if the sperm is already dead or alive by means of modified Bloom's test or eosin nigrosin stain. Next, follow-up testing for a low sperm concentration would include testing for low sperm concentration or sperm count is attributed to problems in transport medium. And again, sino yung organ responsible for the production or secretion of transport medium, which is the seminal fluid? Okay, that is the seminal vesicles. So seminal vesicles may be assessed by means of seminal fluid fructose. Okay. Okay, next, now talk about synovial fluid. So let's talk about its physiology. So the synovial fluid is a highly viscous fluid or liquid found in movable joints or diarthroses. So the highly viscosity characteristic of synovial fluid is attributed to the presence of hyaluronic acid, which is the major mucin or major gel component of the synovial fluid. Okay, so the viscosity is attributed to the hyaluronic acid present in the specimen. So synovial fluid is produced by synoviocytes which line the synovial membrane. So here is the synovial membrane. Here, the humus deep on the synovial joint, synoviocytes. So damage to the articular membranes produces pain and stiffness in the joints, collectively referred to as arthritis. So nasa may articular membrane. So articular membrane or articular capsule is composed of two layers, which is the fibrous capsule, which is the more superficial, and yung synovial membrane naman natin, yun mas malalim. So problems arising with fibrous capsule and synovial membrane can either be through infection or inflammation, may arise, may produce rather arthritis. So arthritis is collective term for group of diseases. So when you speak of arthritis, it doesn't necessarily mean that you have a gout. Okay, so gout is under arthritis. So arthritis is collectively termed or collective group of diseases that arise from damage to the articular membranes or articular capsule. So how will you collect synovial fluid? So you collect synovial fluid by means of needle aspiration by performing arthrocentesis. So the normal amount of synovial fluid is 3.5 ml and the normal synovial fluid does not clot. So again, same with semen, dapat hindi nagka-clot yon. Okay, viscous siya pero dapat walang clot. So to prevent clotting, just in case na nagkaroon ka ng traumatic tap, so syringe may be moistened with heparin. So what are the tubes that may be used for collection of synovial fluid? So for gram stain and culture, you can use sterile heparinized sodium polyanethol sulfonate. For cell counts, you can use heparin or EDTA. Glucose analysis, you can use sodium fluoride. And all other tests, pwedeng non-anticoagulated. Okay, so in here, remember that powdered anticoagulants should not be used because the powdered anticoagulants may produce artifacts that may interfere with crystal analysis. So again, take note, lahat ng to, yung mga anticoagulant na to, in liquid form. Because if you use powdered anticoagulants, it may interfere with crystal analysis. Aside from that, the specimens for crystal analysis should not be refrigerated. So again, because it may produce additional crystals that may interfere with the crystal identification or examination. Next, we have yung color and clarity. So the normal color and clarity of the specimen is that the specimen should be clear and colorless to pale yellow. So it may resemble egg white. So deeper yellow color may indicate non-inflammatory and inflammatory condition. So greenish tinge for bacterial infection. Red, of course, hemorrhagic arthritis or traumatic aspiration. So again, the differentiation between hemorrhage and traumatic aspiration or traumatic tap is that the traumatic tap has an uneven distribution of blood. Okay, so for turbidity, may presence of WBC, synovial cells debris, fibrin, and other cells or inclusion. So how about for milky, may presence naman ng crystals. So again, as I've said earlier, the viscosity of the synovial fluid comes from the presence of the hyaluronic acid, specifically the polymerization of the hyaluronic acid. So how will you evaluate the presence of hyaluronic acids or the polymerization of hyaluronic acid? So you can either perform string test or ropes or mucin clot test. So string test is a simple test to observe the fluid's ability to form, of course, a string. So usually performed bedside sa tip lang ng syringe. So normal, 4 to 6 cm. So for example, less than 4 yung length ng string. So ibig sabihin, mababa yung viscosity, baka may problem sa hyaluronic acid. Okay. So ano ba yung mga conditions na may cause problems in hyaluronic acid polymerization? That includes arthritis. Next form of evaluation for hyaluronic acid polymerization is the Ropes or mucin clot test. So, synovial fluid when added with 2 to 5% acetic acid must form solid clot. Okay, so arthritis lowers hyaluronic acid polymerization, just what I've said earlier. Yung arthritis natin, bababa yung hyaluronic acid polymerization, causing an abnormal result in the Ropes or mucin clot test. That's why instead of a production of a solid clot, the clot is that it becomes less turbid, surrounding fluid that becomes turbid. Okay, less firm, surrounding fluid becomes turbid. So it is not routinely performed anymore because it is non-specific because it only detects the presence of arthritis or lower hyaluronic acid polymerization, but little lang yung diagnostic information pwede mo makuha doon. So nowadays, Ropes or mucin clot test is being used as a means to identify if an unknown fluid is synovial fluid. So if you receive an unknown fluid, if you want to identify if it is synovial, just add 2 to 5% acetic acid. So if it forms a solid clot, then it is a synovial fluid. Next, we have the cell count. So again, WBC count is the most frequently performed cell count. RBC is seldom requested. So pretreatment by adding 1 drop of hyaluronidase again, synovial fluid is highly viscous, and before counting, you should make sure that it is not viscous anymore by adding a proteolytic enzyme, which is hyaluronidase. So manual counts on thoroughly mixed specimens are done using, again, Neubauer counting chamber. So again, WBC count is most frequently performed. So the diluting fluid for WBC count here is not actually acetic acid. Because usually, again, ang general rule of thumb natin kapag WBC count, acetic acid. But here, for synovial fluid, it does not use acetic acid. Because if you add acetic acid to the synovial fluid, that will cause the formation of clots. So again, usually, for synovial, di pala usually, dapat for WBC count in synovial fluid, dapat hindi acetic acid. Because if nag-add ka ng acetic acid, it may cause the specimen to clot. So what will you use instead of acetic acid? So you can use normal saline, or if necessary to lyse the RBCs, okay, for better counting, you can use hypotonic saline or saline with saponin. Okay. So you can also add this hypotonic saline or saline with saponin with methylene blue to further enhance WBC nuclei. So methylene blue, you can also add this one to better visualize the WBC nuclei. How about for cell count normal values? So the WBC count is less than 200 cells per microliter. For RBC is less than 2000 cells per microliter. Next, for differential count. So again, for differential count, still the fluid must be incubated with hyaluronidase. So the normal cells found in synovial fluid includes monocytes, macrophages, and synovial tissue cells or the synovial membrane cells. These are the primary cells seen in normal synovial fluid. So other cells that may be present, neutrophils, of course. Neutrophils may indicate septic condition or bacterial condition. Lymphocytes may indicate non-septic condition. So it can either be through viral infection or inflammation. How about for crystal identification? So for crystal identification, it is important to evaluate arthritis, specifically yung crystal-induced inflammation arthritis natin. So what are the causes of crystal formation? Metabolic disorder, kaya may increased crystals. Of course, that includes Lesch-Nyhan syndrome or alkaptonuria, decreased renal excretion that produce elevated loss of crystallizing chemicals. So may problems sa kidneys. That's why increased yung crystals natin or yung salt content natin sa katawan. Degeneration of cartilage and bones, just what like happens in reactive arthritis. Last is the injection of medications such as corticosteroids. So corticosteroid may cause crystal formation also.